Hello everyone. Good afternoon, good morning, depending on where you are. My name is Karen Berman and I'm CEO of the American Society of University of Haifa. Thank you all for joining us today. Aging, as we all know, is inevitable. It is a reality that will impact each and every one of us. The large and rapidly advancing global population has challenged our research and healthcare communities to decipher the biology that drives aging in order to determine the best approaches for both supporting healthy aging and delaying the process. University of Haifa is proud to be home to the Laboratory of Genetics and Epigenetics of Aging and Longevity as part of our Faculty of Natural Sciences. Our researchers are exploring the mechanisms and pathways involved in aging by examining the biological forces that may influence longevity, proclivity towards certain diseases, and overall health and performance. We are fortunate today to be joined by Professor GIL ATZMON, Dr. DANIELLE GUTMAN, and ASUH Board of Directors member and our longtime friend and supporter, Michael ROSEN. Professor Atzmon is Associate Professor of Epigenetics and Genomics in the Faculty of Natural Sciences at University of Haifa and Associate Professor of Medicine and Genetics at Albert Einstein College of Medicine. His research is dedicated to understanding the association of the whole genome to disease, performance, health, and longevity. To that end, he has established the Israeli multi-ethnic Centenarian study to recruit and profile healthy, independently living Israeli multi-ethnic people aged 95 and older. Dr. DANIELLE GUTMAN is a postdoctoral fellow in the lab of epigenetics and genomics of the aging and longevity headed by Professor Atzmon. As part of her PhD work under the mentorship of Professor Atzmon, Dr. Gutman worked on the Israeli multi-ethnic study, recruiting and processing the samples of over 120 participants. And Michael ROSEN is the Managing Director of Innovation and Research Park and Helix 51 Incubator for Rosalind Franklin University. Michael has founded his own company, ROSEN Bioscience Strategies LLC, a life science economic development consultancy focused on the development of science parks, incubators, and ecosystems centered around research-based universities. He has worked extensively with and trained more than 300 university faculty on the creation of bioscience startups. Michael is also a dedicated advocate of University of Haifa, and we're grateful for his longtime support and assistance. So with that and those introductions, Michael, I'm going to turn things over to you and to our experts.
Thank you, Karen. Well, a very exciting topic today. I have basically dedicated my whole career to the life science industry. And as a baby boomer, I am particularly interested in Gil's comments and research as moving toward this trajectory of aging and the key concept of healthy aging. The University of Haifa, I have been very excited by the developments of the University of Haifa, particularly in the life science arena, although the university is involved in many great areas. But the life science activity has particularly excited me, and with the development of the new Discovery Tower with Rambam Hospital, and all of the life science activity that is going on there that is world-class and having implications for people around the world. So I think we're really fortunate today to have Gil and Danielle give us a perspective on this healthy longevity concept, which is, I think, near and dear to all of us, particularly us baby boomers. And I know Gil is going to start out his presentation first with, or his talk a little bit, with a summary PowerPoint of his seminal work in this area. And then we'll follow up with obviously some questions and put him on the hot seat a little bit, as well as Danielle, to know more about their research. So Gil, I'm going to turn it over to you. I know you've got some great data to share with us on your work.
Oh, thank you. Thank you, Karen, Jennifer, Melissa, and Michael for giving me this opportunity. I would like to share with you a couple of anecdotes that I found during my research on Centenarians and Einstein that have similar feature to our centenarians at Israel. So let me share with you my screen. One moment. Here it is. Okay, moment. Here we are. So the title of this short presentation is 100 is the new 70. And that's what we think about, centenarians. We believe that they have slower their aging process. And by saying that, I will give you some anecdote about it. And before I do so, I want to give an acknowledgement for people who work with me, because this work cannot be done by one person. And I think it's a summary of almost 18 years of research. You can see that for the University of Haifa, there is a lot of names here. Those are my students, and Danielle just finished her PhD. From Einstein, it's Nir Barzilai, Sophia Milman, and Uri. From Rambam, those are the people who help us with recruiting, mainly Tzvi Volevsky, and other people around the world. And of course, the fund, because without money, we cannot do our research. I will start, usually what I'm doing with my audience, real audience, audience that I can talk to them face to face, is to ask which one, is more, let's say, have more impact on the trait of longevity. Is it environment or genetic? So most of the time, people are leaning toward genetics, and this is something that I will convince you that it's wrong. Anyway, so if we look at the genetic perspective, during my time of research, as you can see, it started from 2003 in JAMA and ended and it's still going, but our last paper was in 2017. And we found some candidate gene, meaning genes that are associated with longevity. And if you want to translate it into the global world, you can take those activity of those genes and transform them activity to drugs. And by that, you can treat people and may, they may live longer and better and so forth. By the way, for the CETP, for instance, for example, there is a drug that is now on phase 3 and should be in the market in a couple of years. But if we summarize the whole genetic component that, behind longevity, you can say that the genetic component of exceptional longevity is less than 10%, meaning it leaves us more than 90% for other factors. So if we look at exceptional longevity in healthcare, you can see that the people at the age of 100 spend like 1/3 of people at the age of 70 at the last year of life. Meaning people at the age of 70 spend $43,000 on health treatment, drugs, or whatever it is. And people at the age of 100 spend only $18,000. Again, this is a question to the audience when I'm face to face, and I will just summarize it. We believe they are healthier in every aspect. And I have a slide on it, but in every disease or age-associated disease that you look to them, you can find that they have the same prevalence. It's going by either hypertension, diabetes, stroke, and so forth. So if it's not, let's say, if it's not genetics, it's not health, maybe their lifestyle is different. So when we look at their lifestyle, we look at you can see this from this slide, close to 50% of them are either overweight or obese. I'm talking about the 100 years old guys. If you compare them to the 17 years old guys, you see that they have no difference. Either female or men, they have the same prevalence. So it's not about fat. What about smoking cigarette? What about alcohol? What about physical activity? And what about diet? So they smoke and drink the same as 70 years old, younger control. They don't exercise. They don't have special diet. And almost half of them are either, as I said before, are either obese or overweight. So it's not their lifestyle. So maybe it's the cognition. So in the back-to-back paper, as you can see, 2012, 13, and 15, what we realized that centenarians have personality as well as self-rate health may play important roles in cognitive function. Centenarians also self-report group tend to be optimistic, easygoing, and outgoing. And finally, they have positive attitude, affect, and a favorite personality in conjunction with special rates of health. That might play an important role in mental health outcome. So if we summarize the whole thing, meaning in this case is environment plays a minimal role in exceptional longevity. Maybe the key is the cognitive, but when we look at all other aspects, we see that there is no difference between people who are 100 years old and people who are 70. So if we summarize both the genetic and the environment, we can see that, as I said, the genetic is 10 to 30%. So what's going on with the 70 and 90%? So we think it's the interaction between the environment and the genetic, and this is called epigenetic. It means it doesn't change the DNA sequence, but it's interact with this sequence. And here we can go to the personalized medicine, because each one of us are aged differently. So we need to find out the factor that affects each one of us, understand the portfolio in this perspective, and understand how the environment interact with his or her DNA. And when we do so, we found out that this is the DNA methylation work done by DANIELLE, and she might comment about it later on. What you can see, there is a substantial difference between chronological age, which is the date that is it's put on your birth certificate, and the biological age, which means how you feel, how the body feels, how the system of your body feel. In centenaries, almost 20 years different, and it's very significant. You can see also that in the offspring, we see the same direction, but not as much as a centenarian. In the control, it's going toward this way, but it's not reaching a significant threshold. So if you want to summarize the entire slide deck, our conclusion is that epigenetic may play a crucial role in the way we all age, especially in the health, cognitive, long life demonstrated by centenarians. Thank you.
Gil, thank you. That's fascinating. So let's walk this back a little bit. Tell us a little bit about how you originally got into this field. What led you into this field of research historically? What precipitated your interest in this area?
Oh, that's a long story, and it can last, for decades. But I will start with that I'm a kibbutznik. Kibbutznik is what kibbutznik is. So I was in touch with, farm and soil and good people surrounding me. And then I went to the army, like everyone here in Israel. And then I wanted all the time, studying for my childhood. I was looking to, got interested in biology. So I did my bachelor degree in biology in the University of Tel Aviv. Then I did the master in biotechnology, and my project was the viruses of tomato leaf, or tomato of tomatoes. And my PhD was on chicken, local DNA marker in chicken. And finally, when I went to my postdoc, I came to the lab of near Rosalind, and where we look at the genetic behind longevity. So it's genetic foremost is my interest, and it's depend on the so-called organism that I'm interested in. Then I looked what is the genetic predisposition behind the trait that we look for. And finally, in my last year, when I become an associate professor, both at Einstein and Haifa, I'm looking for the epigenetic. As I said, the genetic component in longevity, it's only 10%. So I guess the larger factor is the epigenetic, and that's what I'm looking for.
So can you define for us non-scientists a little bit, what is epigenetics?
So as I said earlier, epigenetic, it's everything that it's not DNA. Epigenetic, which means, and this is, I call it a new Esperanto, meaning it's language that the DNA speak with the environment, okay? So it's a different element on the DNA that either, giving the gene to express more, express less, or shut down, or whatever it is. But basically, it translates the signals from the world, either the body, the cell, the cosmic signals, whatever it is, it translates it into a language that the DNA understands, and it works accordingly. So for each environment that people is moving from place to place, this translation should be changed and should be depends on the environment.
Great, thank you. So we also have with us today your former student, Danielle. Danielle, can you just briefly just a little bit about your background and how you began to work with Professor Atzmon in this area? What led you to your interest in working with him?
Michael, she's still my student. She's postdoc.
It's hard to leave him. I was also born on a kibbutz, but unlike GIL, I moved around a lot afterwards, living both in Israel and in the States. I always knew that I loved biology since I was a kid, and I was always interested in it. I got married during my undergrad, which I did here at Haifa, biology and math. And during my master's, I had my first wonderful daughter. And during my PhD, I had my second wonderful daughter. So I have an achievement, a personal achievement with every academic achievement. During my master's, I worked on cancer cell lines, and I was doing mechanism, a lot of proteins and imaging of cells and stuff like that. And after finishing it, I knew I didn't want to stay in the field of cancer. And I wanted to get more in-depth knowledge of molecular biology. And I had heard at one of the courses about epigenetics, and that topic really intrigued me because it was very new, and it had power to explain things that genetics didn't. I was lucky enough that at the same time that I finished my master's, GIL joined the university and started his lab and was recruiting students. He gave me the opportunity to work with people as my model organism, because this was very appealing to me, because all the PhD students that I know all work with mice or flies or cell culture as their models. And I had the pleasure to work with doctors and nurses and research coordinators, and most especially these wonderful participants who are willing to take part in our study as a model. These people, I got to, instead of sitting in the lab and dissecting mice, I went and visited 100-year-old people and heard all about their lives. So that was amazing. There were many, many challenges along the way and a lot of frustration, but the good outweighed the bad, and my PhD experience was unique, and I enjoyed every day of it. So that's my background.
Great, thank you, Danielle. So this is going to be a question to both of you, but first I'm going to start with Gil. So Gil, you've had opportunity to be working with Albert Einstein as well as University of Haifa. How can you, and a question to you both is, how do you compare Israeli centenarians versus US centenarians, if you can, on this whole longevity, healthy longevity issue in Israel versus the United States?
The thing is that there is no difference. Meaning, I didn't interact personally with centenarians in US and in Israel more, but in the US it's not, because I'm a guy of you know, data. I'm a guy of genetics. So whatever pull into my hand, either DNA or phenotype or whatever it is, I can see what's the difference. And based on what they experience, what they share with us, what the story that those guys shared with us, we see that there is no difference between the centenarians in the US and centenarian in Israel. Basically, because most of our centenarian is also Ashkenazi. Not everyone is, as I said, most of them are Ashkenazi, so they're both came from the same place. So I guess from genetic perspective, there is no difference. And as I said, we think there is a secret within this guys that they can adjust to any environment they will cope with. Meaning they can cope with any environment they will be. So they act the same, they feel the same, they have the same physiology, same genetic. So that's why there's no, I don't think there is much difference between the two populations, and that is why we combine between them in our reports. In you know, there's a new paper that will come up soon that was led by Danielle, and you will see that there is no much difference between, you know, those centenarians in the US and the centenarian.
Great, Danielle, would you like to add anything to that?
I didn't meet the centenarians in the States. I met their data, and we really are comparing between them now, and we're finding that they're very, very similar. But what I can say about the personalities of the centenarians here in Israel, a great deal of them are Holocaust survivors. And they got to Israel, they immigrated here in various traumatic ways, but they're still very optimistic people. And I can say about all of them, even the ones born here in Israel, that they have this fighter drive in them. These are the people that built our country, and they've watched it develop through the years. So it's pretty amazing to sit with them and talk.
Fascinating, fascinating.
Michael, I want to comment on what Danielle just says. The people here in the US are mostly were born in the US. I think around 70%. So maybe that's the difference. This is Holocaust survivors versus people that was living their entire life in the US.
Great. So just a quick note to our audience. As we are proceeding along, if you've got any questions you would like to forward, on your Zoom screen, you should see at the bottom of the screen a Q&A button, and you can hit that button and type in a question, and we'll try a little bit later on in the presentation to feed those questions into both Gil and to Danielle. I guess if you could maybe summarize, what were the most significant findings in the research, Gil and Danielle, of your research? And obviously, Gil, I know your research has been going on for years in this area, but if you, could pick one particular thing that was the most significant or impact of it, that surprised you.
Well, it wasn't surprise to me, but I think the common denominator, which with every aspect that we, look at the Centenarians, and I have more than 150 papers on it, that Centenarians age slowly. And this is due to better genomic system that adjust to environment changes by epigenome machinery. We try to understand this genetic and genetic makeup in order to incorporate it among the elderly population to increase healthy lifespan alongside with improvement of quality of life. If we can make, this aging process slowly, then we will increase healthy lifespan for everyone, not just those centenarians. But the centenarian has the clue.
Yeah, I have to agree that wherever we look, we see that they're just younger, physiologically younger than their chronological age. And it's highlighted everywhere. So it's amazing.
So one of the questions that's coming through right now actually is very interesting, and it's related to this, is can you tell by age 50 or 60 who is going to be able to, be a centenarian?
That's a nice question, and we ask it as well. So if you are offspring of centenarians, the odds to become centenarians, it's 1 to 9. And in some cases, it's 1 to 17. If you don't have any longevity in the family, so you live like the rest of us. Remember that the being centenarian is 1 to 3,000. The prevalence of centenarians around the world, in the Western country, is 1 to 2,500, 1 to 3,000. So it's rare. It's not that many, but we're going there. We have another paper that says that we're breaking the glass ceiling in order to say, the oldest lived woman was 122 and 128 days. Her name was Jane Clement from France, but we want to break this glass ceiling glass. We want to make it harder. So there is some treatment, some technology, something that is out there that might give us more time to live. But as I said earlier, I don't want just live longer. I want to make it equal the quality of life, that all of us will age gracefully.
So question to both of you. The last 100 days or so, we have been living in this new COVID-19 pandemia around the world. The initial impact of COVID seems to have highly impacted the elderly population. Particularly you see in the United States, elderly nursing homes, assisted living homes, highly impacted by this. My question to you all is not so much how is COVID going to be affecting this population, but how does COVID now impact your research, particularly in this population?
Since I was in sabbatical here at the US and Danielle was at home, Israel, she will testify much more than me. It stopped our research for sure. But the people we recruited earlier before the COVID, surprisingly, as much of them live and protect, most of them live in a protected area, so none of them catch the COVID. Either it's moshavim or kibbutzim they live. And taking what we have so far and going forward, I propose a study to look at those guys, meaning the elderly, and because not everyone have severe symptoms. Some have mild symptoms. And I think that their epigenome profile will predict if they will suffer severely or mildly from the COVID. So if I can take elderly guy, his blood, and see his epigenome profile, I can tell him, you're going to get it hard. So I suggest to you to be isolated, to keep an eye on, to do whatever it needs to be that you won't be affected. But if there is other guys that that it has that I predict that he will get it mildly, I will advise him to do whatever he wants, but keep in mind that it can affect you, but mildly. I'm not saying go and catch it, but you can live more your life not as isolated as the first time. And by learning this dichotomy, I can go further, and not just the elderly. I can go to the younger people and do the same thing and propose the same mechanism. We can predict with the epigenome which drug will affect, which treatment will affect, which treatment will have a good results, and how he or she can recover. So that's the second stage of the COVID-19.
So Danielle, I'm curious, how is COVID-19 impacting your research now in Israel?
Yeah, it pretty much stopped it completely. Since our subjects, it's true that they live in protected areas, but they're also what's called community dwelling. They do not live in old age homes. So even though old age homes were very severely impacted here in Israel as well, our centenarians haven't, because they were under quarantine like everybody else. So they didn't leave their homes. No one came to visit them, and they stayed healthy. But the thing is, because they are at high risk of contracting it, we can't go to them. We can't bring the doctors from the hospital to their houses to sample them. And I don't know when we'll be able to do that again. But we've got to look at the bright side, because this gave us a lot of time to arrange our thoughts, to process all of the results that we had from all of the different methods that we were using to get data. And we are writing papers, and we are thinking of our next steps. What else can we check? What other things, what other directions can we go in based on where those results take us? So it's two sides to the coin.
Right. So in the US, we've seen an acceleration of telemedicine playing a role, a much greater role. And probably one of the outcomes of COVID is the expansion of telemedicine in meeting with patients, both healthy and non-healthy. How is that playing out in Israel now?
Also, we have phone appointments, video appointments with doctors. I believe that Centenarians, they might have, even though some of them were pretty good on computers from what I got to see, but they usually have an assistant living with them that can help them with this, and the doctor doesn't need to physically come to their house to check them. So it's been incredible, the same over here.
I might comment on this, Michael. Those very old guys are looking or aching, let's say, that people will communicate with them. The minute you can do it with Zoom, it's sort of your communication. You don't need to go to their home. The minute you talk to them, you entertain them, it's much of a great health for them. It's, break their isolation and whatever it is, makes them feel good.
Gil, one of the questions we've got coming through is, how does a person obtain his or her own epigenetic profile?
Well, I don't think there is a commercial company that do this epigenome screening, but I can guarantee you that if somebody wants it will be done, he can send it to us and we can do it to him. Although we cannot share the result, we can share the paper, but that's what I can do.
Okay,
There's no
so
commercial
there's
company.
no 23andMe for an epigenetic profile at the moment.
Not yet. I think that it will become in the next year or two.
Okay. So we're getting a lot of questions beginning to come in, and I'm trying to intermix them with some of the prepared questions I have as well as audience questions. So one of the audience questions is, and I'll read this. Can we say that there is a health chasm between our aging process and centenarians? How are technologies being applied to avoid this chasm in lifespan? And how do you use epigenetics to design innovative solutions? Are there immunotherapies being developed in this area?
The answer is complicated, and it's all of the above, what you say. Life expectancy across the world increase every year, although now we speculate that it reached the steady state, it won't go over it. Improve in medicine. Do whatever the doctor tells you. New drugs in the market, new technologies in the market will let us live longer. That's for sure. And it will be in the next 5, 10, or 20 years that life expectancy will increase, I predict, by 10 to 20 to 30 years. My belief are that in the next 30 to 50 years, we can find the mechanism by which we will live forever. But this is, I'm putting aside. The technology that is here today can make us live longer and healthier. It's nanotechnology, it's 3D printers, it's tissue engineering, it's whatever we have in mind, it's there and it's already moving forward. So I guess we will have big news in the next 5 to 10 years. And as I said, living longer, it's easy task. The greater challenge is that to improve the quality of life.
So here's a little bit of a scientific question coming in, and I'm going to need you for our lay audience to explain a little bit. So the question is, can you talk a little bit about your views on epigenetics and telomere shortening and how it affects aging? Do you think that through epigenetics we can try and slow or inhibit telomere shortening? So first of all, what is a telomere?
Telomere is a component on our DNA at the end of the chromosome that protects the chromosome from losing imperative genes. That's its function. There is a mechanism that every time that the cell divides, the telomere becomes shorter. But there is a counteract mechanism which called the Telomerase Gene, which makes the telomere longer. The question is, I have a paper on it that talked about the yin and yang between telomerase activity and telomere shortening. But before I'm going to there, I want my view on telomere length is as a biological thermometer. What do I mean by biological thermometer? It's that it can change every time that you measure. You do right, you eat, either you eat your cereal at breakfast, you have longer telomere, only the one that without the cholesterol thing. You can run or ride a bike or whatever it is. It might change your telomere length. You were angry this day, you were happy this day, it changed. You live your life easy, it changed to the good or changed to the bad. And it's different between people. Depends where live. It's good for the time it's been measured. So if your telomere length is, let's say 37.8 in temperature, it means you have a problem. If it's 36.8, you're good. That's the idea. And you must understand that telomere became shorter with age. Yes, we can fix it, but the problem with fixing it is if you fix it too much, you get cancer. So you need to fix it in slight. So as long as you're on your, the curve, you're fine, you're good. If you're lower than this, you need to seek out what to do in order to improve your life to make them longer. And epigenetic is the same thing because epigenetic can predict what your telomere lengths will be and vice versa. If you're good, if you adapt to the environment and the whole system is working like a good machine, then your telomere lengths will be long. If it's not, it will be a little shorter. That's the idea.
We also have a paper showing that centenarians have very similar telomere length to their controls, meaning, again, a slower aging process, probably more retained telomere length than what you would expect. It's an overrated biomarker for aging.
Mm. So that leads me to my next question would be, what are there a series of biomarkers that you look at that can measure longevity and not just longevity, but healthy longevity? Because that's what our topic is today, not just longevity, but this healthy. Is there kind of a panel of biomarkers that you track or one can track on a regular basis that would indicate not just longevity, but healthy longevity?
Right, so I will move my hat and you can see. So we called it biological clock. So if one of us has biological clock, and this biological clock gives you the interpretation of your physical reaction or physical assessment at the time. This biological function or biological clock is assessed by epigenetics. We started with 300 markers like this, and I think, and we recently published a paper, again, by DANIELLE, the leading writer, and that based on 500 markers or epigenetic markers. And again, with this, we can know for sure what is your biological age as opposed to your chronological age. And there is, equivalent, meaning there is a correlation, because a guy that we predict his biological age is 80 look like 80. And a guy that we predict that is 100 look like 100, every feature that we test. And Danielle can elaborate more on this.
I think we're trying to find biomarkers that will be more personalized. We're not there yet, but the biological marker that Gil was talking about, the biological clock of DNA methylation, is a pretty good one to use, and pretty easy one to use as well.
So just a watch out to our audience, please. Don't forget, we're getting in questions, so please feel free to send in your questions, and we're trying to get to them all. There have been some great questions coming in. So again, please don't hesitate to send in your questions. Danielle, a question to you. You just completed your postdoc, and what are your future plans, and how will your activity with Professor Atzmon continue or change in the near future?
I just completed my PhD. I'm doing mini postdoc at GIL's lab, and the future is very close. September 2nd, me and my family will be arriving in Philadelphia, where I will be joining the lab of Kavitha Sarma as part of the Wister Institute and the Epigenetic Institute at Penn at University of Pennsylvania. I will be working on a new and emerging epigenetic mechanism called R-loops, which is a complicated thing to explain on in a minute. But it's another epigenetic mechanism that contributes to what we see as a whole in a person. And I hopefully will gain enough knowledge on the formation of this mechanism in order to come back to Israel and hopefully rejoin forces with GIL and the whole network that we established here, and try to look at R-loop formation in our older subjects and see how they impact healthy aging in comparison with controls who don't have longevity. So those are my big plans for now.
Fantastic. Mazel tov.
Yes. Thank you.
Gil, question for you. Understand there's a potential for a center for the biology of aging at the University of Haifa. Can you talk a little bit about that?
Actually, this is the basis for me to come to Haifa, because we wanted to establish this center, but didn't happen. But the dream is still there. What I want to build is a multidisciplinary center which incorporates brain research, system and signaling biology, cancer and stem cell research, and of course longevity, which is what I'm doing. We want to decipher aging biology, and that will assist us with essential risk for age-related disease, and it's imperative for advising or advancing our understanding of aging process, age-related disease, and therefore will promote healthy lifespan and aging with grace. What I'm planning with this center is that it acts independently, it will provide state-of-the-art technology, it will focus on basic science and not the nursing aspect of gerontology. We offer translation from bedside through drug therapy, pharmacogenomic, intensive care, personalized medicine. The center will centralize activity, international conference, seminar, and so forth. And it will that associate with the biology of aging. It will foster and initiate our out-of-the-box thinking of undergraduate and graduate students, as well as students from high school in Haifa, meaning approaching the community. In a broader view, the center will encourage new local and global collaboration, will ensure continuity of project that has been developed, and the project and other projects that are in the pipeline. That is it, but you will probably ask why in Haifa, and I will answer that Haifa don't have it. Haifa is the biggest university in the north of Israel. But if everyone in Israel it's the best place for research. This is since they coined the Israel as a startup nation, people understand what it means to do research and doing startups in Israel. So there is no such a thing in the north of the country. And the north of the country, Haifa, as I said, it's the biggest one. It's close to Rambam and Technion that will give people that live longer. And just for your information, Haifa was in the Guinness Book Record for, I think, 3 months for the oldest guy in the world. But then he, left us, so we dropped down. Haifa University is multi-ethnic. It's a diverse population, and it's the diverse population in all Israel, meaning any minority, any Jews and Arab and Palestinian and Bedouin, and whatever you name it, it's there in Haifa. And I think the advantage in doing such a center in Israel is that you can reach in one day from the North University to the Southern University less than 2 hours with Route 6. Less than 2 hours, so you can be in both places in the same day. It's not like here in the US. And since you can do it, the interaction with people, the interaction with great mind, it's much easier, much more fluent. And another thing is the central data of all people that has been hospitalized or in the system in Israel. So everything in center in one place, and you can go there and take whatever you need. I think that's why first why we need center, and second why it needs to be done in Haifa.
Great. So we only have time for a few more questions, and I've got still some audience questions here. So one of the questions that's coming up, Gil and Danielle, is, do you think that with our knowledge of epigenetics and its impact on aging, this will allow humanity to find a way to greatly slow aging? For example, could a 70-year-old still look and feel like a 30-year-old? Or as we say in our title, 100 is the new 70.
So I think I mentioned it earlier. I think that each one of us has its own portfolio. So the more centenarians we have, the more portfolio we will share and we can incorporate in order to make each of one age at the rate we want him to age, and slower is aging. And it doesn't matter if he or she has longevity in the family. The minute we will have his information, we can write down the way it live for the next decades, and slowly and surely, without health problem. And this is based of the centenarian data that we have. Currently, we have a very small one, but I'm sure that we will increase it. And as more as we have of this information for those centenarians, the one that has successfully aged, the more we have information on them, then we can translate it into the broader population. So we need more people.
So perhaps a last question is, for us baby boomers, which represent, at least in the US, more than 72 million people. There's also a baby boomer population in other countries of rapidly aging. I mean, you see rapidly aging happening, obviously, in Japan, Europe, even China. I was startled to see in Brazil, which is generally a country that is typically young, the aging population over 65 is increasing far rapidly, much more rapidly than other parts of the world. What practical advice can you give us on how we can modify our behavior towards this healthy longevity?
So it's 2 points. If you have longevity in the family, you can do whatever you want. If you don't have longevity in the family, please listen to your doctor. And move to Israel. It's the 6th nation in the world for life expectancy. So,
Danielle, any final thoughts on that?
Have really good healthcare for everybody.
Great. Well, thank you. I know we could be going on and on. This is such a fascinating topic and so relevant in all of our lives that I'm greatly appreciative to both Gil and Danielle for this. And thank you for illuminating us on your incredibly incredible research in this very, very important area. So thank you for sharing your thoughts with us today. And I'd now like to turn it back to Karen.
Thank you. I just want to add my thanks to Michael's, to Professor Atzmon, Dr. Gutman, and Michael, to you as well for leading us through this fascinating discussion. As you all can see who joined us, we really only scratched the surface on the science, the impact, and the implications of how we age, and how all of this research can affect our current and future generations. So Professor Atzmon and Danielle, we're going to be looking to you and your work for further insights and maybe some more tips. For those of us, you know, it might be a little late for genetics. We can still listen to our doctors, but if there's any other quick fixes that you come up with, we'd love to hear them. Thank you all for joining us today. We appreciate your time and your interest. If you have any questions that you'd like answered that didn't come up, please send us an email, info@asuh.org. We're happy to pass them on to our experts and have them get back to you. Of course, if you are interested in if this conversation has moved you to support the research and some of the students who are pursuing this topic, we would love your support as well. And you can get more information about that on our website, asuh.org/donate. Thank you again for joining us. We look forward to seeing you all soon. Take care.